Hi everyone, I'm Nigel Gordyke, co-organiser of the Wilmot Terry Fox Run. I'm honoured to be speaking today with Dr. David Malkin in Toronto, who will be telling us about his work. Dr. Malkin is a clinician-scientist and paediatric oncologist in the Division of Hematology, Oncology and Director of the Cancer Genetics Program at SickKids, as well as a senior scientist in the Genetics and Genome Biology Program in the SickKids Research Institute. Dr. Malkin, thank you so much for joining me today.
Thank you very much, Nigel. It's a real pleasure to talk with you.
How did you become involved with cancer research under the Terry Fox umbrella?
So I had started and completed my training in clinical pediatrics and pediatric oncology at the Hospital for Sick Children in Toronto, and then went off to do some research in Boston in molecular genetics and sort of the understanding of cancer biology. And that's how I got really quite interested in the question of cancer and all the problems. And when I arrived back in Toronto after completing that training, actually we have to apply for research grants to support our work. And one of the applications that I put in was to the Terry Fox Foundation at the time, and I was supported by a grant through the Terry Fox for some of the early work, as I was at that point a young investigator, as they call it. And then over the years, we did work with that support and have basically expanded my research interests over the years. But that was sort of the first tangible, if you will, example of the Terry Fox support for the work we do.
Do you have a personal cancer story that led you into this type of work?
So I actually can't say that I do in terms of at the time that I started working in the cancer field in terms of a family story. I mean, there had been family members, like all of us are touched in one way or another, but I don't think that it's fair to say that any of those necessarily drove me to cancer as being an area that I would work in, with two exceptions. So one is I'd always been interested, even before I went into medical school and during, I'd always been interested in doing research. And I was always wanting to do research in a field which was really wide open with many, many questions to be still answered. And back when I was starting, the cancer research field was very much like that. There were far more unanswered questions than there were answered questions. So that intrigued me, that inspired me. I suppose the personal story, if you will, that would sort of drove me, if you will, into cancer was actually not a family story, but when I was an intern, so just coming out of medical school, a first-year intern resident, and I was in the emergency room one evening, and a young girl came in to the emergency room, really quite ill, and the oncologist who was on call that evening, Dr. Mark Greenberg is his name, really has been a mentor since then and still is. He came down to see the child and sort of figure out what was happening, and it turned out that she had a form of leukemia, and he brought me under his wing. He sat with me and we met the family and the young girl together, and I learned and I listened to how he approached this difficult scenario and the plan for treatment. And there was something about that moment, there was something about that point in time that I went back and said, this is what I want to do. And so pretty much ever since that point, that is where I shifted my gears to knowing that I was going to be a pediatric oncologist and I was going to attach science and research to that story.
Could you describe the work that you and your team have been doing? Can you perhaps give us some example projects?
Sure. So yes, there's really two major ones. And the first one starts literally from when I did my training in Boston. I was exposed to some other mentors there, and one of them was a particularly creative thinker. He was a medical oncologist, not a pediatric oncologist, but he was very interested and had spent his career to that point in trying to understand and explore families with cancer. So not individuals within family, but these would be families where multiple members of the family might develop a variety of different types of cancer. And back in the late 1980s, early 90s, there wasn't really a whole awful lot known about why these families developed the cancers that we did. People thought maybe they were exposed to infections or environmental exposures. Genetics was still very much emerging as a discipline. It wasn't as rigorous, or what was always rigorous, but it wasn't as well established as it is now. Sequencing DNA was extremely tedious, as an example. So he had a number of these families that we would see together, and then when I went into the lab there, we thought, along with my supervisor, Dr. Stephen Friend, that it would be actually kind of interesting to see if we could identify the genetic etiology of one of these, we call it cancer predisposition syndromes. And this particular one, called Li-Fraumeni syndrome, is associated with a wide variety of cancers in adults and children, many different types of cancers. Highly penetrant, meaning virtually everybody in the family ultimately gets it if they carry a altered gene. And during the 3.5 years that I was there, we were very fortunate to identify the genetic alteration that causes this syndrome. And that is basically been then, over the last many years that I've run my own lab, the focus of our work. And what we're now doing, leaping forward 20, 25 years, is trying to answer two questions. One is, in families who are genetically predisposed to cancer, can we develop tools that would predict when and in which organ, which tissue the cancer would develop? And can we do that in such a way that an intervention could identify that tumor, and you could actually treat it before it spread or before it became more aggressive? And we've done this in the context of this Li-Fraumeni syndrome, and demonstrated that with relatively simple clinical surveillance tools, blood work, scans, and so on, we can actually do this quite effectively and quite efficiently, and this is actually now used around the world. And that entire area of research has been funded by the Terry Fox Foundation and Terry Fox Research Institute. What we're doing now is saying, could we develop a blood test that might accomplish the same thing, so that patients people don't need to come in constantly getting scans and things, this periodic blood test that would be able to predict. And we haven't figured that one out quite yet. That's where we're aiming for, and hopefully over the next few years we'll find that answer. And then the second area is in the same predisposition piece, is we know people are predisposed to cancer genetically. We've shown that, and many other people have also shown this. Would it be possible to actually prevent that, or at least significantly reduce their risk? Would it be possible to develop or repurpose some sort of a drug or compound that people could take that would significantly reduce their risk? And so that is the concept of, we call, pharmacologic prevention of cancer. And we are embarking on some wide-scale studies with multiple collaborators around the world to explore that question and see where we end up there. So those are two big pieces. And then the last project, which is really quite different, and this evolved over the last few years, is, you're probably familiar with the term precision medicine or precision oncology. This idea of, rather than treating everybody the same way, we recognize we're all individually different, and we should really be focusing on those unique genetic differences that might tell us how to guide management. So we're doing this in the childhood cancer realm. And we have a large project catalyzed by Terry Fox, which is called Profile, Precision Oncology for Young People. And that project is designed at the moment that all children and young adults with hard-to-treat cancers, those that are metastatic, that are have relapsed or have been resistant to treatment, are eligible to have their tumors sequenced using these incredibly detailed sequencing technologies, in the hope to identify molecular events, genetic events in the tumor cells for which then we can actually match specific, precise therapies that may change their course. So that's an ongoing project, and it's a national project. All the pediatric centers in the country and many of the adult centers are participating.
What kind of progress do you think you've seen in the field of cancer research over the years?
So I believe the progress has been actually spectacular. If you look on a day-to-day basis, it's very hard to see that, and I understand that. And for individual families, individual patients with cancer, at a point in time, it can be very difficult to see where that's coming. But if I look back with a wider lens, there are patients that I had cared for back in the say the early 90s when my own career started in earnest. Who had a very grim prognosis based on the type of tumor they would have had. And today, those same patients, if they had lived today with the same disease, would have an outstanding prognosis. And that's in, you know, a quarter century or less. So those have been advances in the way that we design therapy, in the way that we provide supportive care, in the way that we treat the complications of treatment, of chemotherapy and so on, in the way that we utilize radiation, which in children can be very, very debilitating. It used to be when we started, radiation in childhood leukemia was, in most of the common types, was very commonly used to try to protect the brain and the spinal cord. It's almost never used now. With even better results overall. So overall, spectacular progress. However, the challenges ahead are just as challenging now as they were then. The overall survival for childhood cancer today approximates around 82%. It varies depending on what tumor type it is. But for patients who present with metastatic disease or very certain types of cancer, or who relapse, that number drops dramatically. And we still struggle today with how best to treat these patients as we did 20 years ago and as my predecessors did 30 or 40 years ago. So if I put it to the Terry Fox, when he was around 40 years ago, was the anniversary of the run, the treatment that we use now for osteosarcoma, his type of tumor, is the drugs are the same as what he was getting himself. The surgery approaches have changed dramatically. There's lots more to do there. But unfortunately, we have a long way to go in trying to figure out that disease as well as many of the others.
So research is collaborative and requires teamwork. So how has COVID-19 affected your ability to do that work?
So I think it is fair to say that COVID-19 has impacted us, and by us, my own group, but also globally, the national and international cancer research in many ways. The first of all is that the buildings in which we do the research institutes themselves, like everywhere, were shut down for the most part. So a lot of the work that is done is using what we call wet bench research. You're using chemicals and various things at the bench, and you physically can't do that research if you're not physically in the building. So that meant that certain types of experiments simply stopped. The other part is research, like you said, is highly collaborative. We talk with each other. There's corridor conversations. You come up with ideas spontaneously, and this happens just by being in the same environment. And of course, now we're socially distanced, and it is that much more challenging. Having said that, there are also some interesting things that have happened here. So with the use of Zoom meetings or other forms of communication like this, we've been having incredible conversations, meetings, discussions, project planning with colleagues around the world that were really difficult to do before, because you had to somehow or other come up with a time that would work. And we realized, well, hold it, we don't have as many of these other meetings going on. So I just had a call yesterday with a colleague in Cambridge University. We're developing a new project there. as I mentioned, the prevention project that we're looking to explore, we've been actually formally got going on that in terms of the planning stages just about the same time that the COVID shutdown started. And so we are working with multiple groups around the world. We've got things are written, we have a plan in place, and as things gradually open up, we'll be opening that to enroll patients and so on. So a lot has actually happened while we pivoted to a different way of thinking. And then the last piece of it is a lot of cancer research and a lot of geneticists and those of us in the field were able to rapidly pivot certain aspects of our programs to COVID-specific work. And so a lot of the technologies and techniques that one uses in science are adaptable to many things. And so many projects that are being done on COVID-related things are being done by people like us in the cancer field. But having said all that, we all want to get back into the labs as soon as we can, and we are fortunately now across the country, virtually all the research labs are gradually reopening at various stages. And I happen to be in my office today, and just after this call, have a meeting with 3 of my students, keeping socially distanced and wearing masks, because we do wear masks all the time in the building, except when we're in our own office, to discuss some new projects that 2 of my new graduate students will be developing.
That's excellent. So much of your work is conducted in Terry's name. How does that affect you on a daily basis? Is there something about Terry the person that helps to drive your work?
Oh, I think that there very much is. He, you know, he is and still is an inspiring personality. The message of not giving up, of knowing that there is an answer out there, knowing that there is a need to continue to do this work and to continue to work together and to challenge ourselves and to press forward in spite of difficulties such as COVID-19 is really what he was all about in many ways. And so I have a poster of him in my office, and as my door opens, it's the first thing I see when the door opens. And I think that is, I put it there for a reason, and I'm happy that I did. And it does keep me grounded for sure. I've been involved in various aspects of the Terry Fox organization. I do the runs. This year it'll be virtual, of course, for many years. Have been enjoyed to speak to to donors, to the Terry Foxers, to many parts of the program. And I think it is important to recognize that even in times like these, where it is more difficult to fundraise, we have all been hit by financial hardship, that COVID-19 will eventually be something in the past. We will have a vaccine, there will be treatments, and like many of these challenges, at some point it will disappear in terms of the type of challenge we're facing with now. But cancer won't. And cancer is still here. When we open up the window again after COVID, we're still going to have 20% of the pediatric population who's not surviving cancer, much larger portion in the adults. The challenges are still there, and we have to keep up the fight, and that's what Terry Fox was all about.
Dr. Malkin, thank you so much for spending time with us today, and for telling us about the life-saving work that you and your team are working on, and other projects across the country as well, of course, funded by the Terry Fox Run.
80% of children diagnosed with cancer survive, but 1 in 5 will die of their cancer. To get that down to 0, we brought this group of experts together to figure out what is it at a genetic level that makes them so difficult to treat, and find new drugs that are able to target those tumors in a way that we can improve their survival. I'm David Malkin. I'm a pediatric oncologist, a cancer specialist at the Hospital for Sick Children in Toronto, and I'm also director of the Terry Fox Profile Initiative. Currently, the way that children and young adults are treated for cancer are using chemotherapy, surgery, and radiation. And while the success rate of those combinations is very good, the side effects and the late effects can be devastating. What Profile is aiming to achieve is that we are treating each child's tumor as an individual with a precise recipe based on the molecular profile of the tumor, with specific drugs that target only the tumor cells. And in so doing, much better success in killing the cancer and much better success in avoiding all the side effects. And that's the future of cancer therapy. What's unique about Profile is that it is making it possible that any child or young adult, regardless of where they live in the country, will have access to the newest state-of-the-art technologies to do genetic sequencing, and even more importantly, access to a whole array of new drugs which are changing the course of childhood cancer therapy. Our initial goal was to enroll 450 children over 5 years. We've actually now enrolled, when we combined all the parts of the protocol, over 550, and a number of patients who've actually benefited from these new drugs. These would be children who had failed prior therapies where the tumor kept on coming back, and now we've been able to get them onto a drug where we're getting sustained responses and improving their quality of life and increasing their life.